Prokaryotic transcription factors are sort of a mystery when it relates to transcription, elongation, and termination. This is because the RNA polymerase is pretty self-sufficient in handling most everything, so it does not need much extra help. Well, at least in principle. But there are three major transcription factors which could be argued as accessory, but I think they are sometimes very important in either elongation or termination. They are GreA, Mfd, and Nus factors. There are other factors too, but this video will only cover these three. Their main task is to make the RNA polymerase run through obstacles. They prevent arrest or pausing of the RNA polymerase, and can even switch termination to anti-termination events. To understand their function and mechanism of action, I will use the ternary complex, which is also the elongation complex of the bacterial RNA polymerase. I have the initiation and elongation videos explaining how it all assembles. If you need details, check the description for links. We have covered the three main channels in the ternary complex, where the DNA entry channel is the primary one. In addition to them, the fourth channel is at the active site of the enzyme. Imagine this channel to be going through the screen to the back of the enzyme. This is the NTP entry channel, also called the secondary channel. Let's start with the growth-regulating expression factor, or Gre Factor. It serves a positive role in the elongation process. More specifically, it suppresses polymerase pausing or releases it from the arrested state. It also prevents abortive transcription. The suppression of pausing depends on its ability to bypass secondary structures, which means it can restart a paused polymerase. There are two Gre factors, A and B. Their function differs by the length of RNA they can fix. This fixing is usually in the form of RNA cleavage. Before we get into the mechanism, it is good to visualize the Gre factors. Both A and B have similar overall structures. The C-terminal domain binds the RNA polymerase, whereas the N-terminal domains bind to the RNA, which will eventually induce the excision and release the paused RNA polymerase. Ideally, the paused state for Gre to function is some form of secondary structure that has stopped the RNA polymerase. And this is typically the DNA at the transcription bubble. After coming in contact with a paused polymerase, it binds to its beta prime subunit and reaches into the secondary channel. Then, it attempts to push the RNA polymerase backwards. This is called backtracking of the enzyme. To make things more clear, let's focus on the top half of the structure. As the RNA polymerase is pushed back, the mRNA is pushed forward, because the mRNA is bonded to the DNA at the transcription bubble. So as the mRNA comes forward, the RNA moves into the secondary channel, where it eventually finds the Gre factor. The Gre factor then tells the RNA polymerase to cleave this RNA. Keep in mind, the length of backtracking and RNA cleavage depends on the Gre factor involved in this process. The tiny bit of RNA is then released, and the RNA polymerase can restart the elongation, hopefully without pausing. The second factor is Mfd, also called transcription repair coupling factor, because it is involved in the UV DNA repair pathway. The main role of Mfd is also to reactivate paused ternary complexes, and it likes to bind RNA polymerase that are moving quite slowly. It is also involved in recycling the RNA polymerase. That is, if the RNA polymerase cannot be rescued from a paused state. Structure-wise, Mfd is very complicated because it has multiple binding domains and repair domains. In addition, it moves pretty fast on a single-strand DNA and has a non-functional helicase unit. That is because Mfd has homology to recombination family proteins. So Mfd will detect some form of arrested ternary complex, or maybe the polymerase is moving very slowly. The detection of Mfd starts when it binds the duplex DNA at the DNA entry channel. Typically, the distance between the mouth of the channel to the binding location of Mfd on the DNA is around 25 nucleotides. In addition, as you can see, Mfd also binds the beta subunit and has a domain that reaches into the transcription bubble at the active site. Given that the translocase unit of Mfd is active, and it is bound to the DNA outside of the RNA polymerase, it can act like a spring and push the DNA back and forth. Since it is also bound to the beta subunit, it can also push the RNA polymerase back and forth. This is a kind of springy ratchet motion. Essentially, this motion continues until the RNA polymerase wakes up and enters elongation. Otherwise, MFD can also terminate transcription. The last factor on the list is N-utilization substance. The N stands for nitrogen because it was discovered in the context of nitrogen metabolism. Nus factors are multifunctional. They are involved in both elongation and termination. For instance, tryptophan, operon, and some rho-dependent termination mechanisms can be paused by this factor. In other cases of rho-dependent mechanisms and N-utilization regions, it can cause anti-termination and prolong transcription. This requires a bunch of related Nus proteins, and even lambda-phage proteins can cause Nus factors to transcribe viral DNA. Structurally, it is very similar to the Gre Factor. Its C-terminal domain binds the RNA at the nut sites. The N-terminal domain binds the polymerase itself. The important feature of NusA is that it looks like the sigma 70 region 2. We talked about the sigma factor in the initiation video. The region 2 binds near the beta prime subunit, where it melts the promoter and initiates the active site. Therefore, NusA will take its spot during the elongation phase near the transcription bubble and try to interfere with the bubble. Typically, the protein binds RNA at the N-utilization sequence, which is functionally similar to rho-utilization sequence we discussed in the termination video. Essentially, the NusA factor is waiting for the nut sites to be converted into RNA form so it can immediately bind. Sometimes it can bind the site at the exit channel, or if the site leaves the channel, it can also bind it outside of the channel. NusA can recruit other proteins too. The Nus-related proteins can also bind nut sites. They're also lingering near the RNA exit channel, waiting for the nut site to appear in the transcribed RNA. The recruitment of other Nus proteins by NusA to the RNA exit channel can cause physical constriction of the channel, which can then close the RNA exit channel. This force is enough to pause the moving RNA polymerase. In another scenario, it is possible that the nut site leaves the RNA exit channel, like I said before. In that case, the Nus proteins can bind there too. Now, if there was a rho translocase moving to terminate the transcription, the Nus proteins will not let it go further. So now, the Nus factors prevent pausing or even termination of the transcription. This is the anti-termination I highlighted earlier. This is also called read-through transcription because the RNA polymerase failed to terminate. In contrast, Nus proteins sometimes also recruit rho proteins to force termination. Not only that, it can also promote hairpin formation, which can cause intrinsic termination events. As I said, Nus proteins are multifunctional, and it all depends on the context in which the Nus proteins are recruited at the ternary complex. And that is all I have for this discussion.